So, autoimmune disease is a result of a failure of the immune system to recognize self cells. The immune system attacks self cells and then causes some damage. The mechanisms for autoimmune development, though, can be pretty different, as we've kind of seen in the hypersensitivity reactions. Types 2, 3, and 4 of our hypersensitivity reactions lead to what we would classify as an autoimmune disease. It kind of depends on the type of cells that are involved, the location of the antigens, and what cells are actually doing the destruction. So there's a lot there. The development of autoimmune disease is also currently a hot topic in research. Lots of people are trying to understand what's going on here with autoimmune. If we could understand this process too, hopefully we could shut it down as well. Example over here is showing Graves' disease and how Graves' disease leads to the production, through different mechanisms, of antibodies that then bind to the TSH receptor. Instead of destroying this receptor, though, they just bind and kind of block the receptor, or in this case, stimulate the receptor. These TSH antibodies act as TSH, causing to the release of lots of T3 and T4. So this is a hypersecretion of T3 and T4 as a result of an autoimmune disease of antibodies being released from B cells, binding to the follicular cells of the thyroid gland, causing T3 and T4 production. So because this is antibodies binding to a cell, this would be a type 2, type 2 hypersensitivity reaction. So alloimmunity is the ability of the immune system to distinguish that a tissue is not self and then destroy that tissue. The prefix auto in like auto-regulation is self. It means, to act to self. Autoimmunity, destruction of self. Allo, though, means usually different, but associated with the same species. So allo equals different, but still like same species. Alloimmunity comes into play when we receive a tissue from another person or cells from another person. When that happens, our T cells will interact with the donor T cells, or the donor tissues, check their MHC class 1 molecules, determine if it's self or foreign tissues. Now, the MHC class molecule is produced because of several different genes interacting together. MHC class molecules are specific to each individual, but the closer related to somebody is, the more genes you share with them, the more likely that your MHC class 1 molecule is similar to theirs. Even still, though, with siblings and parents and relatives, trying to find a perfect match for a tissue donation is pretty complicated. And even with tissues from closely related individuals, the recipients will still need to take like immunosuppressant drugs. Otherwise, you're going to have rapid graft rejection. And on top of that, even with immunosuppressive drugs, transplant organs still are going to be slowly attacked by the immune system. Depending on the type of tissue, the immune system can destroy it in a few years, or, some tissues like kidneys can last 10 to 20 years, depending on how healthy it was before. But in the end, all donated tissues will be destroyed by the immune system. That process is called graft rejection. For blood, there are specific markers on the cells of red blood cells that help to identify your own cells as yours. It's different than our MHC class 1s. Red blood cells don't have a nucleus, and so technically don't fall into our classification. They don't produce MHC class 1s, but they do have their own sort of antigens on the cell surface, which help us to determine blood types. So there are O types, A types, AB. These different antigens will be presented on the cell surface, and a lot of these antigens are shared between individuals. So a portion of the population are going to have A-type antigens, a portion of the population will have B-type, another portion will have A and B together, some of them will have none, we'll call that O-type. So similar to tissue transplants, when we're giving somebody a blood transfusion, it's important to know what their antigens are on their blood. If you give someone the wrong type of blood, their immune system can attack, their antibodies can bind to their antigens, and that can cause agglutination reactions, or little clumping reactions inside of the blood vessels, which can result to clots forming, destruction of those red blood cells. Those clumps can plug up blood vessels and prevent blood flow. Transfusions reactions can be a big deal. They can actually be lethal. So it's pretty important to make sure that the blood that you're putting into somebody matches the blood that is already there. So those are transfusion reactions. Graft versus host disease is kind of a special circumstance. In this, we usually have individuals who need a bone marrow transplant. So if your bone marrow for some reason isn't working, they'll irradiate the recipient's bone marrow to kill all of the hematopoietic stem cells in there. They'll take a donation from somebody else that's closely related or has similar MHC type 1 receptors, and they'll give that person the injection or the transplant of that new bone marrow. So this grafted bone marrow may have some mature CD4 cells or CD8 cells that don't recognize the recipient's body as self, and so these mature CD4 CD8 cells will go out and start attacking the recipient's body. And because the recipient is already immunocompromised, the recipient's immune system doesn't have the ability to fight these cells off. So you can have CD4 or CD8 immune cells attacking the recipient body, leading to a type 4 hypersensitivity reaction. It's a type 4 hypersensitivity because it involves T cells. So we've mentioned that in some circumstances, the immune system can cause damage to our own tissues. So things like allergies, hypersensitivity reactions, autoimmune disease. The cool thing is, though, the more that we learn about the immune system, the better we are at manipulating it. And so we can use certain drugs, we can use certain treatments to reduce the damage that the immune system can take on our body. Now, in Chapter 3, we talked about anti-inflammatory drugs and some of their mechanisms. We can use anti-inflammatory drugs to reduce the sort of friendly fire that happens in large inflammatory responses. But we got to be careful, though. So anytime that we mess with the immune system, we're messing with our ability to protect ourselves. The immune system obviously exists for a reason, helps to prevent us from becoming infected by things. So messing too much with it, suppressing the immune system, can lead to opportunistic infections. One of the common anti-inflammatory drugs that we mentioned in Chapter 3 are glucocorticoids. Glucocorticoids, things like cortisol, prednisone, dexamethasone, these are really potent anti-inflammatory drugs. They're also steroids, but they act on a wide range of the inflammatory process, like on lots of the signaling cascade molecules. Our synthetic glucocorticoids, like the ones I mentioned, dexamethasone, prednisone, when they're given in a really high dose, usually above whatever endogenous levels glucocorticoids are produced at, they give a completely different response than what our normal glucocorticoids do. So cortisol, which is a normal glucocorticoid, is a stress response hormone. It helps to raise blood glucose levels, free amino acids, get fats into the blood too, so that when your body is stressed, it has the ability or the nutrients available to fight that stress, or at least to be prepared for an attack or to run away or whatever. High levels of glucocorticoids, though, tend to suppress the immune system. Having high levels of glucocorticoids for long periods of time, though, really ramps up that metabolic process, and that can lead to things like thinning of the skin, decreased muscle tone because you're mobilizing proteins, fat deposits in weird places. So it's not a great idea to be on anti-inflammatory drugs, specifically glucocorticoids, for long periods of time. Cytotoxic drugs, we can use these to suppress the immune system. These drugs are pretty potent. They can target rapidly dividing cells like those in the bone marrow. The goal here is to prevent proper cell division. That's kind of the point. By taking these immunosuppressing drugs, you decrease the number of lymphocytes, which will then decrease the effectiveness of the adaptive immune response. So cytotoxic drugs can help with the suppression that way. We can also use drugs derived from bacteria and fungi. These organisms are usually pathogens, and they create chemicals to help evade our immune system. Kind of doing this by blocking some of the cytokines and also kind of reducing inflammation. Drugs like cyclosporin, rampamycin, these are both produced by microorganisms, and they work by reducing our immune response. We can use these drugs to reduce immune response in like tissue transplants too, so these can help with graft rejection. So the body's immune response is there to help attack and defend us. If we can target it, though, if we can like point it at a specific thing and say, hey, attack that, that can also help us to overcome certain diseases. So things like cancer. Cancer, part of the hallmark of cancer is that it evades the immune system. And it does so kind of by ignoring certain chemicals that the immune system gives to it. So like our cytotoxic T cells, if they dump certain granules, enzymes, signals onto cancer cells saying, hey, undergo apoptosis, go kill yourself, certain types of cancers will be like, nah, I'd rather not. But if we were able to develop certain drugs that would help the immune system target cancer cells even better, that could help reduce the danger of cancer just in general. So lastly, we have vaccines. We can use vaccines to stimulate the adaptive immune system. So we inject somebody with a vaccine, a weakened pathogen, and it will produce a primary immune response. It'll produce memory cells, and all of this done to that weakened pathogen. So memory cells float along. They're looking for that pathogen again. The next time we see maybe the real pathogen, the secondary immune response kicks in. This is going to be a greater response, a quicker response, and hopefully that reduces the severity of the pathogen because the immune response is already prepped for it and can remove it before the pathogen has time to multiply and really take hold.